Hey class, today we are doing a yeast lab. I'll be walking you through the setup and you will be collecting and analyzing data. And this will be Unit 0, assignment number 6 in Google Classroom. Now the goals for this assignment, first we want to answer the question, how does a concentration of molasses affect the activity of yeast? So molasses is this, you can buy it at a store, it's this really thick syrupy solution. It's like thicker than maple syrup and it's sugar and it's used to sweeten baked goods. You can either use Peter Rabbit or there's like a grandma's brand out there that is really common as well. So we want to know if I add different amounts of molasses to a yeast, how does it affect its activity? And second, let's practice applying what we learned in Unit 0, Assignment 5, when you took notes on the scientific method. Okay, so our baker's yeast, what we are dealing with, what we will be testing with molasses. Baker's yeast is also known as Saccharomyces cerevisiae, and in society we use it for all sorts of stuff. We use it to bake bread. So anytime you want to make a fluffy baked good, yeast is what you use. And then when you turn 21 and you're able to drink alcoholic beverages, the beer and the wine that you can drink is also made by baker's yeast. So pretty much all over the world, people use baker's yeast in some way. Now, out in nature, it's found on pretty much every living surface. So the surfaces of fruits and leaves and bark and soil, and even in the guts of animals like us, tiny microscopic Saccharomyces cerevisiae is there making a living. Now, what it looks like under the microscope is it's not that exciting. Just a little tiny single cell. And they're one cell. So even we are made of trillions of cells. Saccharomyces cerevisiae is a single-celled organism. And they are most related to mushrooms that you would find growing out of a tree stump or the grass or the soil. They grow in these colonies. So this picture, these are all separate individual yeast, and they grow together in colonies and clumps. And when a single yeast cell, as it's munching on sugar and growing and getting bigger, that one single cell, at some point it decides it's going to divide and form a new yeast cell. So unlike animals, the process of making a new individual is very different with yeast cells than for mammals. It buds. So if you look at this picture, these little like they kind of look like little pimples, little bumps. That's a new yeast cell that's growing off of the mother cell. So the tiny cells are called daughter cells, and then you have the parent cell or the mother cell, the big cells. So for us, what would that look like? Well, if me as a yeast cell, Mr. Gingold yeast cell, came to a point where I had the ability, the resources to make a new Mr. Gingold yeast cell, that Mr. Gingold yeast cell might start growing somewhere on my body. Like, I don't know, maybe on my face. So I have this little cell version of me. This is a brain. And as it grows, it gets larger and larger as it's on my cheek. And it starts, this little baby Mr. Gingold is on Mr. Gingold's cheek. And at some point it pinches off, falls to the ground next to me, gets up, starts collecting food, starts eating, starts growing. It's a separate entity, a separate individual. Now, all these yeast cells, as long as there's sugar, as long as the temperature's just right and there's enough water, they're going to grow, they're going to divide, they're going to bud off new yeast cells. But if conditions get tough, there's no water, it's too hot, it's too cold, there's no sugar around for them to munch on, they form spores. So spores are yeast cells going into hibernation. They change their cell biology so that they work, they move and work really slowly at a bare minimum. It's like putting a computer to sleep. I mean, it's still alive, it can still be turned on, but it's not doing anything. And so when you buy yeast at stores, so you go and you get these little packages of rapid rise instant yeast, okay, these are spores of yeast. They're dried out, they're still alive, they're just not doing anything. They're just sitting and waiting for the conditions to be just right. And so for us, why we use yeast so much, it's not because, oh, it's cool, they like bud off new yeast cells, or oh, how awesome, they're tough, they form spores. That's not important to us. What's important is how they eat sugar. So yeast cells break down carbohydrates and sugars, like the molasses I showed you earlier. And for them, they use that for energy. So they do a process called fermentation to break down sugars into energy. But the byproduct, that's what we like. That byproduct of breaking down sugar through fermentation makes carbon dioxide and alcohol. So when you think of carbon dioxide, if you drink like a fizzy drink like soda, all those little bubbles that are popping and fizzing out of your soda, that's carbon dioxide gas. And that is, yeast produces that as a byproduct. And we do the same thing. If you breathe, you're breathing out carbon dioxide. Carbon dioxide is a byproduct of pretty much all life. When life is breaking down sugars and food for energy, carbon dioxide is a byproduct of it. Hi, class. Right now, I am going to show you some yeast fermentation in action. I have an Erlenmeyer flask. Okay, so an Erlenmeyer flask has a wide bottom, and then it slopes up to the top, and then there's a neck. So I'm going to add a funnel to make sure everything goes in the flask and doesn't get everywhere. I have 100 milliliters of water. I've microwaved it for 30 seconds to make it warm to the touch. Yeast do better between 80 to 90 degrees Fahrenheit. So I've microwaved the water. And now I'll add it to the funnel. There we go. I also have 20 grams of table sugar. So I'll add that. So now I have my solution. Let's mix it up. So the table sugar will act as a food source. Mix it up, make sure it's mostly dissolved. And then I have some Fleischmann's rapid rise instant yeast. So the yeast that you buy from the store, this rapid rise, the yeast, it's alive, but they're in spores. So they've gone into hibernation because they've been dried out so they can survive on the shelf for a long time. So by adding the water and the sugar, I'm going to wake up the yeast. So I'll pour a whole packet in, about 7 grams. All right. I do not need my funnel anymore, so I'll set that aside. I will mix this up. So as the yeast senses the sugar and the water and that 85 to 95 degrees Fahrenheit temperature, the yeast spores should start to activate, and they should begin fermentation, begin breaking down the table sugar into carbon dioxide. So I'm adding this balloon to capture carbon dioxide gas. We can't see carbon dioxide gas, but we can see it fill this balloon up. So if this balloon fills up, we know the yeast, it's alive, it's fermenting, it's grabbing that table sugar, ripping it up, breaking it apart into energy and carbon dioxide gas. So I'll give it one final mix, and then we will play the waiting game. I've let 2 minutes pass. I paused it. You don't need to stare at a screen for 2 minutes looking at a balloon rise. But you can see, I'll note a couple things. The balloon, it's got some air in it. It's like a little mini punching bag. That's carbon dioxide gas being produced by the yeast. If you look as well, you can see it's starting to get a little foamy. So carbon dioxide gas is getting trapped in the water and the sugar and is forming a frothy layer on top of the yeast mixture. And I don't know if you look, you can maybe make out bubbles being formed in the yeast solution. Oh no, my balloon popped! Put it back on. I just experienced some experimental error, human error on my part. The balloon popped off and then I ripped it while I tried to put it back on. So now I have a green balloon. And in a matter of a minute, it has reinflated itself. And you can see as we speak, I don't know, this balloon may be a bad balloon to use because it looks like it's inflating right here. And you can see that the foam is increasing, all that carbon dioxide gas being trapped in water and sugar and making this little foamy, gassy matrix. The yeast in the water is fermenting away, breaking down that table sugar into carbon dioxide gas and alcohol and energy. And I want to try to hold on so you can really see it going now. Yeah, so it's fermentation. So this process right here is the same process that makes bread rise in the oven. So I'll pause here and then I'll come back on to show you a bigger balloon. So the balloon's popped off. I'll give up on the balloon, but you can see that the carbon dioxide matrix of foam is rising, and pretty soon it's going to be out of that Erlenmeyer flask. And one last time, there is, if you can see it, you can't see the yeast because they're microscopic, but you can see the carbon dioxide bubbles being made, and you can see the foam rising out. So now I need to put this somewhere so it doesn't get all over my table. And so fermentation at a, well, to make it a little more complex, and this is something we'll learn about in more detail later on in the semester, this is the biochemical pathway of fermentation. So basically, we just want to pay attention to the beginning and the end. So we start out with glucose, a sugar cube. Okay, here's the sugar that I would use to power up my yeast. And I add something called ADP, and I have some enzymes breaking down that sugar. Well, it releases energy that the cell can use. It also releases carbon dioxide gas that makes the yeast bubble that filled up the balloon. And then it also produces ethanol. Now, if you're thinking, oh, so if I use yeast to make bread, I can make the bread alcoholic? No. When you're baking bread, the ethanol just evaporates away. So, sorry. Hey, class. So right now we are going to set up our yeast lab. I have all my materials out in front of me. So first, I have this green test tube rack to hold my test tubes. All right, I have 5 16 by 125 test tubes. Now the 16 means it's 16 millimeters in diameter, 125 millimeters high. And then I have 5 13 by 100 millimeter test tubes. All right, I have 2 graduated cylinders. So they're graduated because they have all those lines that help us measure volume. I'll be using 2. I have my 10% molasses solution. I have a suspension of active dry yeast. I have parafilm. So parafilm, parafilm is scientific grade saran wrap. So it's stretchy and you wrap it around stuff. Creates an airtight seal. I have a plastic pipette tip. I have water. I have 2 plastic forks for stirring. I'm using this Erlenmeyer flask to capture my waste. And then I'm using 2 200 milliliter beakers to hold my solutions. Okay, so let's start. So first, I labeled my test tubes 1, 2, 3, 4, 5. So I'll start with 1. And so I want to put 15 milliliters of my 10% stock solution of yeast. So let's move this over here. So I have my yeast, or my molasses, I mean. So let's pour 15 milliliters in. And so when you're measuring, you want to be eye level with your graduated cylinder. Okay, so there is about 15. I need to dump a little bit out. Yep, so there's 15. So in test tube 1, I add 15 milliliters. Perfect. All right. So now in test tube 2, I want to start diluting the molasses. I want different concentrations of molasses to test my yeast in. So number 1 acts as our stock, our baseline. For 2, I want to dilute it by half, by 50%. So starting with 10%, I'm going to add 25 milliliters. So let's get to eye level and make sure I'm even. Looks good. I'm at 25. Now, to dilute it, I'm going to add 25 milliliters of water. Okay. So now here, I want to add 15 milliliters to my test tube. So I'm going to measure it in my other graduated cylinder. Need to dump a little more. Okay, so I'm measuring at eye level. I have 15 milliliters. Okay. So now with the remaining amount, I want to dump. I want 25 left in here. So I have 35. I need to dump 10 out. So I'll just dump here. Okay. So I'm keeping the 25. The 10 I dumped in here, I will dump into my waste container. I'll put my waste container in here. Okay. So now, to this 25 milliliters of molasses stock solution in water, I'm going to add another 25 milliliters of water. Okay, and so if you see, take a look at the color compared to what we originally started with. So now I want to add 15 milliliters of this molasses and water solution to test tube 3. I have 15 milliliters. I'm adding to test tube 3. There we go. So I'm going to dump 10 milliliters out of my stock solution. Okay, so dump this in my waste. And now again, to this molasses water solution, I'm going to add another 25 milliliters. So now we can, let's add, now add 15 milliliters. I'm going to add to test tube number 4. I added a little bit too much. Okay, so then I'm going to dump 10 milliliters out and put it in the waste. And then one more time, I will add 25 milliliters of water to my molasses and water solution. And I want to add 15 milliliters of this water and molasses solution to my final test tube. All right. So I have test tube 1, 2, 3, 4, and 5. Each time I added 15 milliliters, I diluted 25 milliliters of the molasses and water solution with 25 milliliters of water. And I actually am going to add a test tube. I'm going to rinse this out. I'm going to add 15 milliliters of water to a 6th test tube. There we go. Okay, so I can put this aside. Now, I need to add this smaller test tube. Now, I want to add it not face up, face down. And I do not want any air bubbles. So right now it's sticking up. So to make it so that there's nothing but liquid inside of that tiny test tube, I flip it over. So you can see that it went down. You might have to do it a few times tight seal with my thumb. Okay, there's that. So I will do it to the remaining ones. All right, now I've added the 13 by 100 test tube upside down into all 5 test tubes. I shook it upside down, each one, so there was no air in the bottom of the test tube. So now I need to add the yeast. So I want to mix up the yeast solution. So I have it here. Give it a good mix. I'm using a plastic fork. You can use anything really to mix it up with. I have a plastic pipette dropper, so I'm going to pull out a pipette full. And I want to add 10 drops to each test tube. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, so I've added the yeast. And the final step, I want to add some parafilm. And so parafilm is going to seal the test tube. Pull it off. So I'm going to rip off a piece. And again, it's nice and stretchy. So I'm just going to stretch it over the top of the test tube. So with a tight seal and go upside down. Over my laptop. That's how much I trust this parafilm. And nothing comes out. So it's sealed. Now that I have all 6 of my test tubes, the tops are sealed with parafilm. I'm going to let these sit for 24 hours. So at the end of 24 hours, we will come back and we will measure how much carbon dioxide gas is made in each test tube to see how much activity the yeast had in the molasses.